Welcome back to yet another new session on Bioinformatics. Today our topic would be Sequence Patterns and Profiles Part 2. Before moving to the session, we shall see some of the key words and their definitions. Motif: conserved regions of protein or DNA. Motifs often contain important features. Pattern: qualitative description of motif based on regular expression like syntax. Profile: quantitative motif description using weight matrix syntax. Sequence motif discovery based on conservation. Sequence motifs are conserved sequences of similar or identical patterns that may occur within nucleic acids, DNA or RNA, or proteins either within different molecules produced by the same organism or within molecules from multiple species of organisms. In the case of cross-species conservation, conserved motif indicates that a particular sequence pattern may have been conserved during evolution to perform certain functions. Thus, motif conservation is the basis of motif discovery by studying similar genes or proteins in different species. A motif discovery program that considers phylogenetic conservation is named phyloGIPS. Based on alignment, this process is usually done by first constructing a local alignment of multiple sequences, after which the highly conserved regions are isolated based on their high alignment scoring. After the highly conserved regions are isolated, they are used to construct profile matrices for each conserved region. The profile matrix for a given motif contains frequency counts for each letter at each position of the isolated conserved region. We can extract the so-called consensus sequence, that is, the string of most frequent letters. Without alignment, the motif start positions in sequences can be represented as the set S, S1, S2, S3 up to ST, where ST is the position index. Scoring motifs: given S equal to S1 to ST, we align T motifs of length L from all sequences. Construct the profile matrix. Motif score would be 3 plus, 4 plus, 4 plus, 5 plus, 3 plus, 4 plus, 3 plus 4, which equals 30. Motif score S for DNA is sigma max, count Ki. Here count Ki represent the frequency of nucleotide K in the motif starting at Si. Tools for motif discovery and searching. Protein domain prediction. Examples of programs predicting specific domains. CIPRED, protein sequence analysis workbench including secondary structure and disordered protein prediction. Phoebus, transmembrane helical segments and signal sequences. Coils, prediction of coil-to-coil regions characteristic for structural proteins or proteins involved in transcription regulation. MEME suite. The MEME suite web server provides a unified portal for online discovery and analysis of sequence motifs representing features such as DNA binding sites and protein interaction domains. The popular MEME motif discovery algorithm is now complemented by the GLAMP2 algorithm, which allows discovery of motifs containing gaps. 3 sequence scanning algorithms, MAST, FIMO and GLAMP2 scan allow scanning numerous DNA and protein sequence databases for motifs discovered by MEME and GLAMP2. Transcription factor motifs, including those discovered using MEME, can be compared with motifs in many popular motif databases using the motif database scanning algorithm TomTom. Transcription factor motifs can be further analyzed for putative function by association with gene ontology (GO), terms using the motif GO term association tool GOMO. MEME output now contains a sequence logos for each discovered motif as well as buttons to allow motifs to be conveniently submitted to the sequence and motif database scanning algorithms MAST, FIMO and TomTom or to GOMO for further analysis. Major databases. Some examples of conserved domain databases include ProCite, www.expressi.ch/ProCite/. ProCite is a database of protein families and domains. It consists of biologically significant sites, patterns and profiles that help to reliably identify to which known protein family, if any, a new sequence belongs. It was started by Amos Bayrock, is part of SwissProt and is maintained in the same way as SwissProt. The basis of this is regular expression describing characteristic subsequences of specific protein families or domains. ProSite has been extended to contain also some profiles which can be described as probability patterns for specific protein sequence families. The site can be used to search by keyword or other text in the entries, to search for a pattern in a sequence or to search for proteins in SwissProt that match a pattern. Scan ProSite. Scan ProSite is a tool to scan a database with ProSite or user built patterns. PFAM. PFAM is a database of protein families defined as domains, contiguous segments of entire protein sequences. For each domain, it contains a multiple alignment of a set of defining sequences, the seeds and the other sequences in SwissProt and Tremble that can be matched to that alignment. The database was started in 1996 and is maintained by a consortium of scientists, among them Erik Sonhammer, CGB, Sweden, Sean Eddy, Washington University, St. Louis, USA, Richard Durbin, Alan Bateman and Ivan Burney, Sanger Center, UK. The alignments can be converted into hidden Markov models HMM, which can be used to search for domains in a query protein sequence. The software HMMER by Sean Eddy is the computational foundation for PFAM. The domain structure of protein sequences in SwissProt and Tremble are available directly from the PFAM websites, and it is also possible to search for domains in other sequences using servers at the websites. The PFAM database can be searched or used to identify domains in a sequence or downloaded from the websites above. The PFAM database is licensed under the GNU General Public License, which basically makes it available to anyone but imposes a restriction that derivative works, new databases, modifications must be made available in source form. Prints, provider of fingerprints, groups of aligned, unweighted motifs. Blocks, a database of weighted profiles or blocks. These databases all have different areas of optimum application. It is difficult to assure which of them gives best results. They all have a particular strengths and weaknesses. Profile based database searches using PSI BLAST and analysis and interpretation of profile based searches. Introduction to PSI BLAST. A powerful tool for searching sequence databases with a probe sequence is PSI BLAST from National Center for Biotechnological Information (NCBI). PSI BLAST stands for Position Sensitive Iterated Basic Local Alignment Sequence Tool. It can be accessed from the URL www.ncbi.nlm.nih.gov/blast/psiblast.cgi. An earlier program, BLAST, worked by identifying local regions of similarity either through local or global alignments. In global alignment, both sequences are aligned along their entire lengths and the best alignment is found. In local alignment, the best subsequence alignment is found. The PSI in PSI BLAST refers to enhancements that identify patterns within the sequences at preliminary stages of the database search and then progressively refine them. Recognition of conserved patterns can sharpen both the selectivity and sensitivity of the search. PSI BLAST involves a repetitive or iterative process as the emergent pattern becomes better defined in successive stages of the search. PSI BLAST provides an automated facility for constructing, refining and searching PSSMs within the context of a single program. Starting with a query sequence provided by the user, the process begins with a standard BLAST-P search of a sequence database. Highly significant alignments found in this search are then used to construct a PSSM on the fly. Comparisons of the PSSM against the sequence database are performed using a variation of the word-based BLAST algorithm used for standard sequence comparisons. The process continues until no new matches are found or a specified limit or number of iterations is reached. Many functionally and evolutionarily important protein similarities are recognizable only through comparison of three-dimensional structures. When such structures are not available, patterns of conservation identified from the alignment of related sequence can aid the recognition of distant similarities. These patterns are variously called motifs, profiles, position-specific score matrices and hidden Markov models. In essence, for each position in the derived pattern, every amino acid is assigned a score. If a residue is highly conserved at a particular position, that residue is assigned a high positive score and others are assigned high negative scores. At weakly conserved positions, all residues receive scores near 0. Position-specific scores can also be assigned to potential insertions and deletions. The power of profile methods can be further enhanced through iteration of the search procedure. After a profile is run against a database, new similar sequences can be detected. A new multiple alignment, which includes these sequences, can be constructed, a new profile abstracted and a new database search performed. The procedure can be iterated as often as desired or until convergence when no new statistically significant sequences are detected. The design of PSI BLAST. Iterated profile search methods have led to biologically important observations, but for many years were quite slow and generally failed to provide precise means for evaluating the significance of their results. This limited their utility for systematic mining of the protein databases. The principal design goals in developing the position-specific iterated BLAST or PSI BLAST program were speed, simplicity and automatic operation. The procedure PSI BLAST uses can be summarized in 5 steps. A. PSI BLAST takes as an input a single protein sequence and compares it to a protein database using the gapped BLAST program. B. The program constructs a multiple alignment and then a profile from any significant local alignments found. The original query sequence serves as a template for the multiple alignment and profile whose lengths are identical to that of the query. Different numbers of sequences can be aligned in different template positions. C. The profile is compared to the protein database, again seeking local alignments. After a few minor modifications, the BLAST algorithm can be used for this directly. D. PSI BLAST estimates the statistical significance of the local alignments found. Because profile substitution scores are constructed to a fixed scale and gap scores remain independent of the position, the statistical theory and parameters for gapped BLAST alignments remain applicable to profile alignments. Finally, PSI BLAST iterates by returning to step 2 an arbitrary number of times or until convergence. Profile alignment statistics allow PSI BLAST to proceed as a natural extension of BLAST. The results produced in iterative search steps are comparable to those produced from the first pass. Unlike most profile-based search methods, PSI BLAST runs as one program starting with a single protein sequence, and the intermediate steps of the multiple alignment and profile construction are usually invisible to the user. PSI BLAST practical. The method used by PSI BLAST involves a series of repeated steps or iterations. PSI BLAST is most conveniently used on the internet with the help of the graphical user interface provided by the PSI BLAST search page on National Center for Biotechnology Information (NCBI) website. First, a database search of a protein sequence database is performed using a query sequence. Second, the results of the search are presented and can be assessed visually to see whether any database sequences that are significantly related to the query sequence are present. Third, if such is the case, the mouse is clicked on a decision box to go through another iteration of the search. The high scoring sequence matches found in the first step are aligned, and from the alignment a type of scoring matrix that indicates the variations at each aligned position is produced. The database is then again searched with this scoring matrix. Thus, the search has been expanded to include sequences that match the variations found in the multiple sequence alignment at each sequence position. The results are again displayed, indicating any newly discovered sequences that are significantly related to the aligned sequences in addition to those found in the previous iteration. Again, an opportunity is given to go through another iteration of the program, but this time including any newly recruited sequences to refine the alignment. In this fashion, a new family of sequences that are significantly similar to the original query sequence can be found. To demonstrate the improved sensitivity of the PSI BLAST approach, the sequence of histidine triad HIT protein was used as a database search query. Baxvanis and Hewlett, 2001. With a standard single pass BLAST-P search, no significant hits or no putative conserved domains have been detected. However, with a multi-pass search, new relationships to GAIT are discovered at each iteration as shown in figure below. Similarity between HIT and galactose 1-phosphate uridylyl transferase was later described based on superimposition of their three-dimensional structures, Holman-Santer, 1997. However, sequence similarity between these two proteins is extremely weak. The RAT-GAL-T protein is found in the second iteration, and the information from this alignment is incorporated into the profile. Several additional homologs from other organisms are also identified. PSI BLAST versus BLAST. Because of its cycling nature, PSI BLAST allows to find more distant homologs than a simple BLAST search. PSI BLAST uses two e-values, the threshold e-value and inclusion e-value. The threshold e-value is used for the initial BLAST and the default value is 10 as in the standard BLAST. The inclusion e-value is to accept sequences in the PSSM construction and the default is 0.001. Now, let us have a quick look at what we have discussed in this module. This module explains the basic concept and definition of sequence patterns, motifs and profiles. Various types of pattern representations are discussed and an attempt has been made to demonstrate the profile based database searches using PSI BLAST and analysis and interpretation of profile based searches. Before we conclude, please try to answer the following questions. A. Find out the protein family to which the signature sequence P-LIVM-C-T-LIVM-X-F-T-P belongs to using scan-prosite tool. B. Develop a motif for arginine deaminase enzyme and represent it in the prosite syntax. C. Prepare the flowchart of the steps followed in a PSI BLAST search. Here are some books for your reference. EMB Net Course Basel by Marco Pagni, Lorenzo Cerruti and Lorenza Bodoli, Swiss Institute of Bioinformatics. Bioinformatics Methods and Applications, Genomics, Proteomics and Drug Discovery by S.C. Rastogi, Parag Rastogi, Namita Mendirata. Computational Methods by G.R. Liu, V.B.C. Tan, X. Han. Bioinformatics, a practical guide to the analysis of genes and genomes by Baxvanis A.D. and Hewlett B.F.F. 2001. Thus, we conclude today's session. See you soon with another module. Thank you.